Hello, biology. The purpose of this video is to show you some of the types of genetics problems that you are going to have on the genetics problems quiz, and additionally give you an opportunity to see the types of questions you're going to see on the multiple choice portion of that. Of course, because this is a video and you can pause to go look things up in your book or on your study guide, I will be going a little bit faster than I normally would in the class. But again, if you are using this as an opportunity to study and look up some things and confirm what you know or what you don't know, you'll need to pause the video at some points. What we're going to do is we're going to do one category at a time, and we'll start with vocabulary. Genes that mask or are stronger than others are said to be blank. If two plants are big T, and big T, little t, they have the same what? Answer in 5, 4, 3, 2, and 1. Okay, so if they mask the other one or they dominate the other one, they are dominant alleles or dominant traits. And big T, and big T, little t, if we're talking about short and tall plants, will both have the tall phenotype. They have different genotypes, but the same phenotype. A capital R represents a dominant allele, and the lowercase r represents a recessive one. What would be the genotype of the hybrid? The principle of blank states that some alleles are dominant and some alleles are recessive. The genetic makeup of an organism is called its what? About 15 seconds. Answers coming in 5, 4, 3, 2, and 1. Okay, so a hybrid is going to be a big R, little r. You might also see or hear the term carrier. That would mean that this is carrying the allele for the big R or carrying the allele for the little r. And in most cases, a carrier is referring to a recessive allele. Principle of dominance states that some alleles are dominant and others are recessive, and the genetic makeup or the letters or the codes for the traits is the genotype. The chemical factors that determine traits are called what? The symbol or symbols or letter or letters used to represent a recessive trait is or are what? So I'm not telling you if it's one symbol or two symbols. Which scientists used hybridization to make a new line of plants? Traits that are produced by the interaction of several genes are called what? I apologize in advance if you can hear me slurping my coffee, but it's relatively early in the morning and I'm not quite awake yet. Answers in 5, 4, 3, 2, and 1. Okay, chemical factors that determine traits are called genes. You might also see the term alleles. Two lowercase letters would be the recessive trait. That is the only way the recessive trait would be shown, is if there is no uppercase letter with a lowercase letter. Luther Burbank was the guy who used hybridization to make new plants, and polygenic many genes. All right, on to some genetics problems and some random questions. If pea plants have two different alleles for the same trait, they are said to be blank. Being tall is dominant to being short. A man heterozygous for tall and a short woman have children. What is the probability in a fraction that their kids will be short? I'm going to take a little bit longer on these problems as I'm going to work out the solutions and show you the actual solution. All right, man's heterozygous. Woman short. All right, answer coming in 5, 4, 3, 2, and 1. All right, so heterozygous, if they have one allele for tall and one allele for short, and then the chance is 1/2. And let me show you how that problem is solved. Okay, 5 steps. Identify the letters and the phenotypes. What are the parents? Remember, when you have a duplicate allele that comes from the same parent, you can cross it off. It makes our Punnett square a little less complicated. And of course, this one's short, this one's tall, 1/2 chance. All right, here we go. Heterozygous plant and a homozygous recessive small plant are crossed. What is the probability in a percentage that the dominant trait will be seen in the offspring? What is the probability in a fraction that the recessive trait will be seen in the offspring? All right, answer coming in 5, 4, 3, 2, and 1. All right, so 50% chance for the dominant, 1/2 chance for the recessive. If this problem sounds familiar, it's because we had one that was very, very similar to that. So I'm using the previous solution and just simply changing. Give me 1 second. My Elmo's not cooperating. Come on now. There we go. Okay, so it's the exact same setup. 50% chance that it's going to be tall, and it is a 1/2 chance of seeing the recessive. But the setup and everything else is correct. I just simply changed this part. Okay. Okay, 2 trait cross problem. Brown hair is dominant to blonde hair. Being short is dominant to being tall. A heterozygous short man with blonde hair and a tall woman heterozygous for brown hair have children. What are the possible phenotypes? And what is the probability in a fraction that a child will have brown hair and short? All right, answers coming in 10 seconds. Five, four, three, two, and one. Okay, so you're going to have all four possible phenotypes. Short person with brown hair, short person with blonde hair, tall person with brown hair, tall person with blonde hair, and of course, the probability of one of those is going to be 1/4. And here's how that problem was solved. Okay, so identify what the different traits are. You have heterozygous or homozygous dominant for short, homozygous recessive for tall, heterozygous or homozygous dominant for brown hair, homozygous recessive for blonde. Step 2, identify what the parents are. Step 3, indicate what the possible alleles are. Notice I only indicated 2 for each because there are going to be duplicates. Anytime you had a homozygous, you're going to get to cross them off. So I crossed off 2, crossed off 2. That left me with 2. Plug them into a Punnett square. These are the 4 possible phenotypes, and it's 25% for each. All right, here we go. We're going to skip genetics problems. We're going to come back to those later. All right, homologous chromosomes, sex chromosomes, and autosomes are all shown in a blank. Gregor Mendel used pea plants to show the blank of traits or characteristics. Answer coming in 5, 4, 3, 2, and 1. All right, all those can be seen in a karyotype. And he used pea plants to show the inheritance of traits or characteristics. Blank is the likelihood that a particular event will occur. The visible or physical characteristics of an organism are called blank. An organism that has the same genes for a particular trait, big A, or little a, is said to be blank. All right, answer coming in 5, 4, 3, 2, and 1. All right, so probability is the likelihood that a particular event will occur or not occur. Visible or physical seen characteristics are phenotypes, and this is going to be homozygous dominant or homozygous recessive. All right, units of heredity are called blanks. True or false, Mendel used pea plants because they only have a single visible trait, which makes them easier to study. When Mendel crossed true breeding tall and short plants, all the plants were blank because the allele for blank plants is dominant. If a Punnett square has big T, and big T, little t, the offspring will be blank. Answer coming in 5, 4, 3, 2, and 1. All right, units of heredity are called genes, traits. Either of those would work. Mendel did not use pea plants because they had one trait. They had a variety of traits, so that is false. True breeding, tall and short plants, all of the plants were tall because the tall gene or allele is dominant. Remember, they were all big T, little t, and he was probably scratching his head going, what the heck just happened? Because I know I used a seed from a short plant. Okay. Let's see. If a Punnett square has big T, and big T, little t, the offspring are all going to be tall. Okay. Or the offspring are all going to show the dominant trait. It's going to be big T times a big T, homozygous dominant. Big T times little t, heterozygous, still dominant. Big T times big T, homozygous dominant. Big T times little t, heterozygous, but still dominant. All right, non-Mendelian genetics. When black and white parent chickens produce a speckled chick, this is an example of blank, when you can see both of the traits of the parents in the offspring. What's that called? Human skin color differences are example of what? Five, four, three, two, and one. All right, so when they're both seen or they're seen together, co means together, that's codominance. And human skin color, human eye color, human hair color, they are all polygenic traits. Multiple genes interact to make those phenotypes. True or false? Big B, and Big B O both result in the same phenotype. So these are blood types. Most sex-linked traits are found on the blank chromosome. 5, 4, 3, 2, and 1. That's true. This is B-type blood. This is B-type blood. It just simply has a heterozygous situation with the recessive O-type blood. And most X-linked traits are found on the X chromosome. If a couple has 3 boys in a row, what is the probability and the percentage that their next child will be a boy? How many pairs of chromosomes are found in humans? 5, 4, 3, 2, and 1. Every time you have a new child, you have a 50% chance of having a boy or a girl. It does not matter what you had previously. Now, if I asked, what is the probability of having 3 boys in a row or 4 boys in a row, that's a little bit more different because that is 4 events that are dependent upon one another. So in that case, you would actually have to multiply the fractions. But each individual event is 50% or 1/2 chance. And be very careful with this. 23 pairs is equal to 46 chromosomes, not 46 pairs. 23 pairs, 22 autosomes, 1 through 22. And then, of course, you have the sex chromosomes. Here we go. All right, on to the random problems. In the P generation, a tall plant was crossed with a short plant. Short plants reappeared in the F2 generation because the allele for shortness and the allele for tallness blanked when the F1 plants produced gametes. Down syndrome is also referred to as blank 21. Answer coming in 5, 4, 3, 2, and 1. All right, so they separated or segregated. Remember, the only way to get that recessive allele away from the dominant allele so it could be paired with the other recessive allele is if you separate them during gamete formation. All right, and Down syndrome is referred to as trisomy 21 because there are 3 copies of chromosome number 21 for Down syndrome people. The law of blank states that 1 gene or allele from each pair goes to each sex cell. Didn't we just talk about that? In humans, chromosomes 1 through 22 are autosomes. What is the last pair of chromosomes called? The principle of blank can be used to predict traits of the offspring produced by genetic crosses, and a blank shows all the possible results of a genetic cross, the genotypes of the offspring, and the alleles in the gametes. Five, 4, 3, 2, and 1. Okay, so it's the law of segregation. X and Y chromosomes are the sex chromosomes. It's the principle of probability can use to predict traits. And a Punnett square is a tool that we use. It shows the results in a genetic cross, the possible results, the genotypes of the offspring, that's going to be inside the Punnett square, and then the alleles in the gametes, that's going to be on the outside of the Punnett square. And this one should have been a 4-point question. That change. All right. Okay. This one I need to change as well. All right, here we go. All right, Alyssa and Mike, her brother, have brown hair, which is a dominant trait. Alyssa's son does not. Actually, I don't think you do not have a pedigree on. No, we're not going to do this one. I think this link is broken, actually. All right. Okay, on to the genetics problems. I'll need to make that change later. Okay, color blindness is a dominant sex linked trait. A woman who is a carrier for the trait and a man who has normal vision have kids. What is the phenotypic ratio expected in the children? Okay, so. All right. Woman's a carrier. Man has normal vision. Okay, what is the phenotypic ratio expected in the children? Okay. All right, we've got a. Sorry, I'm talking to myself. Okay. All right. And here, so it's a 1-to-1 to 1-to-1. So you have all four possible combinations. Let me show you how that was solved. Okay, so big C, or big C, little c is colorblind. Homozygous recessive would be normal. Mom is a carrier, okay, which means she has one big C and one little c. Dad is normal, which means all he needs is one little c. All right, those are the four gametes. Plug them into a Punnett square. So obviously, both of these are going to be females. This one is colorblind. This one is normal. Both of these are going to be males. This one is colorblind. This one is normal. So that is a 1-to-1 to 1-to-1 ratio, one representing all four possible. And of course, these are the symbols for females, and these are the symbols for males. All right. Okay, a woman with AB blood and a man homozygous for A blood have children. What are the possible phenotypes of the children? What is the genotype of the woman? And what are the two possible genotypes for A type blood? All right. Answer coming in five, four, three, two, and one. All right. So it's going to be A, B, or A blood. My bad. I'm changing this. Get less confusing. Okay, here's what that looks like. Okay, A blood can be big A, or big A O, heterozygous, and that is also the answer right here. All right, mom is AB. These are her two gametes. Dad is AA. You get to cross off of one. All right, plug them into a Punnett square. A type blood, AB type blood. Kids can be either one. Mom is AB. Okay. Rolling tongue is dominant to not being able to. A woman heterozygous and a dad who cannot roll his tongue have kids. What is the phenotypic ratio expected in their children? Second genetics problem. White and red flowers are incompletely dominant to one another. Red flowers crossed with a pink one. What is the genotypic ratio expected in the offspring? All right, answer coming in 5, 4, 3, 2, and 1. All right, so we got a 1-to-1 ratio. It'll be 2-to-2 if you don't actually cross off the duplicate allele. And let me show you how to work those problems. All right, so first one. Kid you not, this new Elmo is frustrating. Okay. All right, so mom is heterozygous. Okay, so we're going to have a big R and a little r as the gametes. Dad cannot roll, so he's homozygous recessive. And of course, we like it when they're homozygous because that means we always get to cross off one of the gametes. Here's mom's 2 gametes. Here's dad's gametes. We get a heterozygous. They can roll their tongue. We get a homozygous recessive. They cannot. That's going to be a 1-to-1 rolling tongue to cannot roll tongue. All right. Here's the incomplete dominance problem. Incomplete means incomplete in between. So the pink is going to be the heterozygous. WW white, RR red. Doesn't matter what letters you use. All that matters is that you get the phenotypes associated with the combinations. One of the flowers is red. That means we get to cross them off because they're homozygous. Okay, RW we cannot. RW and R, we end up with a red. We end up with a pink. 1-to-1, the genotypic ratio is going to be the big R to big R, big W. All right. And I've made a decision, even though you do not have a pedigree chart on your test, we're going to do this problem anyway. All right, so Alyssa and Mike, her brother, have brown hair, which is the dominant trait. Alyssa's son does not. Draw a pedigree chart. And what is the husband's genotype? Okay, and this one we're going to get rid of because I've already got that on another problem. And we're going to make this 3-point question still. Okay. Okay, answer coming in 5, 4, 3. Oh, my bad. This is supposed to be word 2 now. Okay. All right, so let's see. So this is going to be Mike. This is going to be Alyssa. All right. It is a dominant trait, so both the brother and sister have the actual trait. All right. We know that because the son does not have the dominant trait, they have to be homozygous recessive, which means that Alyssa has to be heterozygous. and we're not exactly sure of what the husband is because the offspring does not have the trait. So could have got the little b, okay, from had to get the little b from both parents. All right. Actually, no. This one, if it was little b, it could have got either one. If it was big B, little b, it still could have got the little b. Yeah, so we don't know what this parent is. Okay, so as long as you got the phenotype, pardon me, the pedigree chart correct, you got that 1 point. And let's get rid of this question. Get rid of this question. Right, so as long as you got the phenotype as unknown correct, you got 1 point. And as long as you got the pedigree chart correct, you got the 1 point. All right, ladies and gentlemen, I hope you study hard and do well on the test. And I think we have finished all of the questions. All right, y'all, we will see you in the class.